This is chapter 5, and so we're going to get started here with the very first topic in chapter 5. So chapter 5 is about all organic molecules, which we call large biological molecules, meaning they're all going to contain carbon as their base. And to build these molecules, we're going to start with small pieces and join them together. The small pieces are called the monomer, and when you chemically bond one monomer to another, that is a polymer. Okay, so for each one of these types of molecules, for carbohydrates, lipids, proteins, and nucleic acids, we're going to discuss the monomer, and then we're going to discuss the polymer. This transparency here, this picture, figure 5.2, is showing you how the polymers are formed. This is a particular chemical reaction. It's known as a dehydration synthesis reaction. Dehydration because water molecules are coming out of the reaction. Notice how the arrow is pointing out. That means it's leaving the chemistry of the reaction, and it's forming this new chemical bond here between the short polymer of 3, and we're adding another monomer to form a longer polymer that now has 4 pieces. The opposite reaction, when we break the polymers apart, is called hydrolysis, because hydrolysis, we're using water to break down the molecule. So notice the water molecule is drawn now with the arrow pointing into the chemical reaction. The arrow symbolizes the chemical reaction. And notice one of the hydrogens from the water is going to join monomer 3 here at the end of the polymer, and the hydroxide portion, the OH, the other hydrogen and the oxygen, are going to rejoin the unlinked polymer. If I want to continue to break this polymer down, I'm going to need to add a new water molecule to break each of these bonds. You might have learned this dehydration synthesis reaction. An older name for it is a condensation reaction. So you may have learned that in chemistry as well. If you have taken Chem 102 or you're currently taking Chem 102, much of this will probably be familiar to you. Okay, so now let's talk about the first group that we need to talk about. So I'm going to get rid of this. Wait a minute. Whiteboard, clear this off. Okay, and I'll just use my text button here. Yeah, let's make it blue, why not? So the first group of organic molecules that we need to talk about are carbohydrates. And shorthand for carbohydrate is CH2O, right? And that H should be, I can't do that on this whiteboard, but that should be, right? It should be drawn like this. Got my pen back here. So it's carbon, and then twice as many hydrogens as oxygen and the carbon. Okay, so that's a shorthand for carbohydrates. The monomer for carbohydrate, so the building block for carbohydrate is called a monosaccharide. And I will type that rather than try to write it. Right. And examples of monosaccharides, I'm going to show you some pictures in a second, are molecules. Usually for the monosaccharide, we say between 3 and 6 carbons. And of course, if there's 3 carbons, then there would be 6 hydrogens and 3 oxygens. Right? These monosaccharides can be drawn either in a linear form, or once you get to 5 and 6 carbons, they will be drawn in a ring form. So I'm going to go back and let's take a look at, I don't like it does that. Let's take a look at, let's take a look at some monosaccharides. I am not interested in you knowing which ones are aldoses, ketones. I do not care, right? A lot of this is irrelevant. What I want you to look at and focus on here is how many carbons, how many hydrogens, and how many oxygens. Okay, this is a 3-carbon carbohydrate monosaccharide glyceraldehyde. We're going to see this one a few times as we go through these molecules. That's why I'm pointing it out. The pentoses here, the 5 carbons and the 6 carbon sugars, we're going to see ribose over and over and glucose over and over. Okay. Notice these are the linear forms. As I mentioned, once we get to 5 and 6 carbons, they're going to form in water. They're going to automatically form this ring form. And you guys do need to take a look at this picture because you do need to know how the carbons are counted, and you need to know what this abbreviated picture looks like. And it's going to be even further abbreviated, and they're going to take the numbers out. Okay, so this is glucose, which is one of the monosaccharides. This is glucose in its linear form, and they're showing you step by step how the ring is formed. I'm not going to ask you what bonds are made here, but you do need to be able to recognize that this is a monosaccharide. Do you have to tell me this is glucose? Absolutely not. But if I have this picture on the exam, you need to say carbohydrate monosaccharide. So here are the carbons. Notice they've abbreviated them, which means they've taken out drawing the C for carbon here. And the implication is anytime you have 4 chemical bonds here, that means there's a carbon there. So this is the first carbon, the second one, the third, the fourth, the fifth, and see how the 6th sticks out bonded to the 5th carbon here. We count the carbons clockwise after the oxygen in the ring. That's why this is carbon 1 and this one is not. This is carbon 1 because it's after the oxygen clockwise in the ring. 1, 2, 3, 4, 5, 6 carbons. Single ring, monosaccharide. Functions in the cell. Functions of monosaccharides, 2 functions. They are a source for cells to produce energy. And remember, in cells, the form of energy is chemical energy. It's called ATP, all capitals. And the second function is to be a monomer, to join together to make polymers, right? So let's take a look at some polymers of carbohydrates. These are 2 examples. They occur via a dehydration synthesis reaction. The 2 polymers we have here are called maltose and sucrose. Most of the carbohydrates, the monosaccharides and disaccharides, this would be a disaccharide because there's 2 monosaccharides end in OSE. And notice there's a dehydration synthesis, water's coming out, and usually what I tell students for the visual clue for a polymer of carbohydrates is you're looking for rings containing just carbon, oxygen, and hydrogen, and those rings are holding hands with an oxygen. No, you do not have to know the 1,4 linkage, the 1,2, don't care. I need you to be able to see this picture and say carbohydrate, disaccharide. Functions of disaccharide, energy. That's what they're used for, energy in cells, make ATP. Now, when these polymers get much longer, we call them polysaccharides, and those are going to have different functions because they are polymers. Here are some examples of polymers of glucose. Starch, glycogen, cellulose. These are different polymers of just glucose. Depends on how they're joined together. So here's some starch granules inside some potatoes. Here's some glycogen. Notice they're showing you that in muscle tissue in a person because animal cells produce glycogen as a way to store carbohydrates. And cellulose. So this is a structural form of this particular polysaccharide. So if I would go back to my whiteboard that I was using before, if I can, and I would type in here polysaccharide, And again, if you have the note outlines in front of you, yeah, you don't really need this because it's already on there, right? So the functions of polysaccharides, there would be two. One is to store energy, and remember that's in the form of ATP. And the second function of polysaccharides is structure. And what I mean by that is cell walls, particularly in bacteria, and plants, right? So this is a cellulose fiber that is going to become the cell wall, this yellow part of a plant cell, right? So storing energy in the form of starch and glycogen, different forms in plants and animals, and structure. So polysaccharides have two functions. I think these are just smaller, bigger pictures of all of them separately, and you can see these pictures in your textbook as well. Here's just the chemical, what it looks like if we were just looking at the monomers joined together to form the polymers. Again, am I going to show you this picture and ask you to tell me that's starch? No. If I show you this picture, you have to say that's a polysaccharide, which is a type of carbohydrate, and it can be used for storing energy or structure, like in the cell wall. Here is a more unique polysaccharide. It's a polysaccharide called chitin. Notice that it has the base glucose ring, but it has this nitrogen component hanging on here. And this in a polymer actually forms the exoskeleton of arthropods, and it forms the cell wall in fungus. So again, structure. These would be more examples of a polysaccharide that is structural. That's what you need to know about carbohydrates. The second group in your book is lipids, which I'm glad they moved to lipids there. In earlier editions, they had the proteins there, and I always did the lipids second anyway. So let's take a look at lipids. I'm going to add to my whiteboard here and put another text box here and put in lipid. So lipid category is a little more complicated. We will discuss three subcategories of lipids: fats, phospholipids, and steroids. Okay, they all share something in common with their structure. All has something in common. We're going to look for glycerol, so that's a carbohydrate. Plus, in the case of fats, we're going to see plus fatty acids. That's the basic structure of all the lipids. As I mentioned, we're going to look at three specific types of lipids: a fat, phospholipids, and steroids. All right, let's see if I can just, how do I get back to my other picture? Maybe just doing that. Okay, there we go. Okay, so let's take a look at a lipid. This is a fat, and fats are glycerol, which is a carbohydrate, 3-carbon sugar, plus 3 chains of fatty acid. And a fatty acid is, we call this a hydrocarbon chain because it's carbon with 2 hydrogens, right? Notice the fatty acid chains are joined to the glycerol again by a dehydration synthesis reaction. And in the case of a fat, which is also known as a triglycerol or a triglyceride, we have the glycerol molecule and 3 chains of fatty acids. These fatty acids do not have to be the same length of carbon. You can see how there's, I don't even know, 9 carbons here, maybe, and only 7 here. There it is bigger, here it is again, right? So a fat is one type of lipid and comes in 2 varieties in that we call them saturated and unsaturated. Saturated means every single one of the bonds in the fatty acids between the carbons is a single bond, meaning they're only sharing 1 pair of electrons. Unsaturated fats have a double bond. And usually what that double bond does is it causes a bend in the molecule, a shape change. That's why most unsaturated fats are liquids, like olive oil, and saturated fats can be packed closer together, and at lower temperatures, they'll be solids, like butter. Just pictures of each of them. Second type of lipid that we are going to discuss is called a phospholipid. Put a big yellow star on this picture, okay? This picture is going to haunt you till the end of the semester, okay? Phospholipids, this is the structure first, and then we'll talk about the function. So structure first, a phospholipid. So it has a phosphate attached to it, which is from chapter 4, this molecule here. Notice the glycerol is sideways here, and we have 2 chains of fatty acids. One of them is saturated, one of them is unsaturated, so it makes this little kick or kink in the molecule. We have the phosphate, and then attached to the phosphate is a choline group that is very charged. Gives this head of the phospholipid a very negative charge. The head we call hydrophilic. Because of the charge, it's going to be attracted to water because water is polar covalent. The tails are fat and they are hydrophobic. They do not like to bond with water because there is very little charge here. When phospholipids are together in a group, they, without any interference, they form a bilayer. This is the function of the phospholipid. Get ready, write this out. Function of phospholipid, all membranes in all cells are composed of a phospholipid bilayer. And that is so important that I'm going to have to add that to our list here. All right. So we talked about fats. And now we're talking about phospholipids. And that is super important that under the phospholipid you type all membranes. I can't spell. In all cells are composed of a phospholipid bilayer. Now, there are other things in that bilayer, which we'll talk about. We have a whole chapter about this. Chapter 7 is all about the membrane, right? This is the basic unit of the membrane. So for fats, we looked at the structure, glycerol, 3 fatty acid chains. Function of fats in cells is to store energy and provide insulation. Phospholipids, got glycerol, 2 chains of fatty acids, phosphate group, and their function is to be the membrane, all membranes in all these cells. All right, let's go back. And the last type of lipid that we're going to talk about today is a modified lipid. This one is cholesterol. It's a steroid, or sometimes if there's an OH group on here in another chemical place, we call it a sterol, right? This is a modified lipid. So it contains glycerol and a chain of fatty acids, and then these 4, you can always recognize a steroid because it will have these 4 rings. Notice how the rings aren't holding hands with oxygen. The rings are sharing carbon bonds, and some of them are double bonds. This is the clue of how you recognize a steroid. And the function of steroids in cells, function of cholesterol, is to help maintain the shape of the phospholipid membrane. And obviously there are also steroid hormones. So they would be involved in chemical signaling, particularly the sex hormones that direct the development of the reproductive parts of primarily animals. In plants, we see the steroids sometimes as chemical signals as well as structure. And you would see them in the structure of fungus membranes as well, different sterols. Okay.